Is it started? Okay. So we are now going to talk about the elements that make up the electron transport chain. So as I mentioned last class, there are many proteins that form these complexes. There are 4 complexes that make up the electron transport chain, many proteins as part of each of these, but those proteins do not handle electrons. There are specialized molecules that are embedded or incorporated in these proteins that allow for electrons to move through these complexes in a series of redox steps. So it's a little bit, it's been referred to as a molecular chain or a molecular wire that allows for these electrons to be moved. And we're going to consider for just a few minutes the various different kinds of elements that we will see. So we have talked about FAD before and FADH2, and that molecule is represented on the left. It is this ring at the top that is able to accommodate electrons. These electrons are received one at a time, bringing about ultimately FADH2, the fully reduced form of FAD. But we will also see something called a flavomononucleotide, which does not have the AMP attached to it. So just keep in mind that the FMN that we will see behaves almost identically to the FADH2, but it is truncated, so it looks different, its size is different, the place where it will be bound is going to be different in these complexes. The other thing I would mention is that FAD or FMN, neither one of these can leave the protein complex. So while they're able to receive electrons, donate electrons, they cannot move and carry electrons away from the complex. We talked about NAD plus, NADH. This is the NAD plus molecule. Again, it is this ring atop the ribose there that is going to accommodate the donation of a hydride with its 2 electrons. All right, there are many copies of a molecule called ubiquinone or ubiquinol, depending on its oxidation state. It is, as you can see, it has this long hydrophobic chain that sticks off one end of it. This ensures that it is embedded in the membrane. So this ubiquinone, is an electron shuttle, and it moves between complex 1 and complex 2 to complex 3. So it collects electrons, then moves them to complex 3. It's able to handle 2 electrons, and because of its sort of aromatic nature, the semiquinone, the free radical form of this, is somewhat stable. But in the end, the ubiquinone is going to gather 2 electrons. It's going to release 2 electrons to complex 3. It actually moves through the inner membrane. So it actually is part of that rather fluid lipid bilayer. We're going to see a series of heme groups. So these are protoporphyrins with iron present. These are going to be able to accept and release 1 electron. And this becomes important as we consider the electron transport complex 3. We've seen the protoporphyrin 9 before. It's the same ring that is used in hemoglobin. Here we will see it in B-type cytochromes. So we will see some heme Bs, we'll see some heme Cs, and we will see some heme As in different complexes of the electron transport chain. And we also, again, will see iron sulfur clusters. We saw these as part of the conatase. There, it was part of the active site and was able to stabilize the intermediate. But here, these have the sole function of moving electrons through a protein complex from a donor molecule such as NADH to a recipient molecule such as ubiquinone to become ubiquinol. So there are single iron, we have 2 iron sulfur clusters, 4 iron sulfur clusters. These are all part of these complexes. Yeah. No, you won't need to know the structures of these until the exam. No, just all right. Now the remarkable thing is that as the electrons move, okay, and we're going to see that there are, even within a single complex, there are going to be a series of steps. Each of these represents an oxidation reduction. Each time an electron moves from one iron sulfur cluster to the next, it's a redox reaction. Overall, these reactions can only take place if there's a release of energy. So we're releasing energy every time, well, not every time, but we are releasing energy overall as we move through one complex, or the electrons wouldn't move. So even though we're using the same elements, sometimes the protein that surrounds them is able to change their overall redox potential to allow for a reduction in energy as we move electrons through each complex and from one complex to the next. All right, so this outlines the 4 complexes. The first of these is by far and away the largest. 43 proteins make it up. There are a series of elements that handle the electrons. We'll talk about these individually. But in the end, those electrons are transferred to ubiquinone to form ubiquinol, which will carry those electrons on to complex 3. Complex 2 is succinate dehydrogenase, step 6 of the citric acid cycle. We've seen this before. In this particular instance, the electrons are first transferred to FAD to form FADH2, and through a series of iron sulfur clusters, again, releasing those electrons to ubiquinone to form ubiquinol, which is carried on to complex 3. Complex 3 has 11 proteins. It has a series, it has 1 iron sulfur cluster, but it has a series of heme groups. One of these hemes is a C heme, 2 of them are B hemes. In this case now, the electrons are passed to a molecule called cytochrome C, which is present in the liquid between the outer and inner membrane of the mitochondria. And this cytochrome C will carry 1 electron at a time from complex 3 to complex 4. Complex 4, with its 13 proteins, is where oxygen undergoes reduction to water. So in the overall metabolism of hydrocarbons, proteins, fats, we end up with carbon dioxide and water, but it doesn't all happen in a single step in our body. The water is produced in the final complex of the electron transport chain. This one has some interesting properties. It has some copper centers. It has some heme groups. And we'll talk a little bit about how it may bring about the reduction of oxygen to water. Okay. In the end, the point of all this is to actually pump protons. It seems crazy, but all of this energy that is being released as part of these series of oxidations that is part of each complex, that energy is harnessed through mechanisms that we don't understand to pump protons from the matrix to the intermembrane space. This creates an energy supply, if you will. It's a little bit like putting water in a water tank high above the ground. It creates then a resource that's going to be tapped into for ATP synthesis. So here is complex 1. For one NADH that is reoxidized, 2 electrons enter, pass through a series of components, and in the end, we pump 4 protons from the matrix to the intermembrane space. So, we can see these then moving out here. You'll notice that over on the right, we have the ATP synthase, and those protons are actually going to re-enter through the ATP synthase. Remember now we have an increase in concentration, so we have a disequilibrium. We have an increase in positive charge on the outside of the inner membrane. This is going to create then also electrochemical energy that can be used to generate ATP. Succinate dehydrogenase, the second complex, does not pump any protons. Complex 3, for 2 electrons that pass on to cytochrome C, again pumps 4 protons. The complex 4, for 2 electrons coming in, we have 2 protons coming out. Okay. All of these protons then re-enter through the ATP synthase, and that's how we are going to generate ATP. Okay, so let's consider complex number 1. 43 proteins. We have a binding site for NADH. So this NADH is also called the NADH dehydrogenase. It is going to reoxidize NADH. The NADH is coming from the pyruvate dehydrogenase complex, much more abundantly from the citric acid cycle. But this is where the NADH molecules, with their electrochemical energy, are going to donate those electrons. Initially, they're going to go to this flavomononucleotide. So we have this flavomononucleotide right here. And then we have a series of 9 iron sulfur clusters. Now, the book talks about 6 to 9. Actually, I think there are 9 in virtually all species. The issue is how many of those are used as part of this chain. So we have this molecular wire where we are moving electrons along in static X-ray solved structures of the complex 1. Some of these iron sulfur clusters seem too far away to participate. However, I am of the opinion that they wouldn't be there if they weren't going to be used. And so in an actual viable cell in aqueous conditions, under normal physiologic function, that probably all of these iron sulfur clusters handle those electrons. Now think about this. Each of those steps represents a redox step. And overall, the energy in the system has to get lower and lower, or there is no free flow of electrons. Ultimately, those electrons are conferred to Q to form QH2. Q being ubiquinone, sometimes called coenzyme Q. If you go to the drugstore or your local supermarket to the supplement aisle, you'll find this if you want. I'm not sure how much of this actually gets to you, but that's what it's used for. You can see this ribbon structure down below here. You can see that these iron sulfur clusters are positioned quite close to one another, allowing the electrons to move freely through this. For 2 electrons entering, 4 protons are pumped. They're pumped by this complex, and they move from the matrix to the intermembrane space. How this is accomplished is not fully understood, but it involves conformational changes that open channels to accept protons from the matrix side, conformational change as electrons move through the series of iron sulfur clusters, opening up channels that deliver protons to the intermembrane space side. Yes?
Do we know what happens to the electrons?
Yes, they're given to Q and become ubiquinol, and then they are moved to complex 3. Okay? Yes?
How come 2 electrons allows 4 hydrogens to come through? We don't know.
Yeah, so the stoichiometry of this is really not. It's not so much the number of electrons, it's the amount of energy that is being released overall through that series of electrons. That is sufficient to be able to cause 4 protons to be moved to the intermembrane space. You'll need to remember that you're moving things against their gradient, okay? So they already have too many protons in the intermembrane space, but you're pushing more protons into that same space. Even more importantly, there's a charge. There's this positive charge buildup on the outside of the intermembrane, and you're pushing more positive charge into it. And so it takes a substantial amount of energy to bring about the movement of the protons into the intermembrane space.
And it's just purely the electrons that are providing that energy from
Yeah,
molecules?
yeah. So each of these components that handle the electrons are tuned in just the right way to allow for enough energy to be released to pump those protons. Yeah. So it's not a simple process. Okay, let's move on to complex 2. Let me just highlight it here. Well, this is maybe not the best figure for it, but here it is. So it is shown here. This is step 6 of the citric acid cycle. It turns out that succinate dehydrogenase is not soluble in the liquid in the matrix of the cell, but it is membrane bound. And so the electrons move from succinate. In doing so, we form fumarate, the oxidized product. Those electrons are transferred initially to FAD and then on through they're moved here to a series of 3 iron sulfur clusters. Okay, this is a little bit harder to see, but here is the FAD. Now remember, the FAD is permanently embedded in this complex. This complex is made up of 4 proteins. Complex 1 had 43 proteins, so this is a much simpler complex. Those electrons, so the substrate binding site is right down here somewhere, according to the arrow. The electrons move here and then on through 3 iron sulfur clusters onto Q, ubiquinone, to form ubiquinol, and that moves through the intermembrane onto complex 3. There's also a heme group there. It is not known whether it participates in this electron transport or not. It would be surprising if it's not. It's not that easy to build a heme and then embed it in a particular protein at where it is, but nevertheless, it seems to be far enough away from the iron sulfur clusters that it's not clear that it actually receives the electrons. This complex, unlike complex 1, 3, and 4, does not pump protons to the intramembrane space. Okay. Complex 3, let me go to, this is, we're going to walk through this, but this contains 11 proteins. It contains hemes. It contains one iron sulfur cluster that is shown up here. It's called a Rieske protein. The iron sulfur cluster is part of that. We see a C heme. We see some B hemes over here. And the electrons are going to be transferred ultimately. So we're going from QH2, ubiquinol, the reduced form. We're going to transfer those electrons through complex 3 to cytochrome C. There is a problem here. The problem is that the QH2 is bringing in 2 electrons, and the cytochrome C can only accommodate 1 electron. And you have to bring in then an additional copy of the oxidized cytochrome C to pick up a second electron to carry it off to complex 4. And so it turns out that there are 2 separate pathways. So those 2 electrons arriving with QH2, one goes one way and the other goes the other way. And so I'm going to actually go to the board and talk a little bit about this. So here's the problem. We have QH2 with 2 electrons interacting, docking with complex 3. And over here, we have our cytochrome C binding site. And this can only accommodate 1 electron. This provides 2 electrons. And there is no simple way for the electrons to be housed anywhere. I mean, you can't simply stop their movement. Their movement is extremely fast. So if you think of electricity, it moves at a pretty high rate of speed. So QH2 comes in, and it's going to, 1 electron now is going to go to an iron sulfur cluster. It's transferred immediately here. Then it's going to be moved to a heme group. So we're going to have heme C1. Okay? And this heme is embedded in a protein, called a cytochrome protein, and it is part of the complex. This heme now will pass that electron onto cytochrome C. Once this is reduced, it will move on to complex 4. But this takes time. On a time scale, the movement of electrons is much, much faster than the ability to bring an oxidized copy of cytochrome C in, pick up an electron, move on, bring in another copy of oxidized cytochrome C to pick up the second electron. So that's why there needs to be a second pathway that allows for the second electron to be moved immediately elsewhere. So we're going to have now 2 heme groups. Oops. BL, BH, And interestingly, they are going to give these electrons to a fully oxidized copy of ubiquinol. Okay, so here's reduced ubiquinol coming in on this side. Over here, there's a second binding site that will bind oxidized ubiquinone. So this electron comes in, it produces now this free radical, which is, because of the aromatic nature of the ubiquinone, is able to stabilize that for a period of time. So this is now one electron to cytochrome C, moving on to complex 4, one electron to this Q, and then we repeat the process. We're going to, so this gets oxidized, reoxidized. We're left with this, so now this cytochrome C comes off, moves on to complex 4, and now we bring in a second copy of oxidized cytochrome C, and we bring in another QH2. This inner membrane is full of Q in QH2. There is a reservoir of these molecules. They are able to pick up these electrons and move them about very efficiently. So again, this is the second copy now. Let me just mark it as number 2. It brings in one electron, which is going to now reduce a second copy of cytochrome C, which is going to go off to complex 4, and another electron, which will come through this arm and bring about the full reduction of this. This now comes off and joins this pool of QH2, which is present in the inner membrane. It can come back and dock with another copy of complex 3 and deliver its electron. So in the end, we are going to oxidize 2 copies of QH2. We're going to reduce 1 copy of Q to QH2. And we're going to reduce 2 copies of cytochrome C, one at a time. And they will carry 2 electrons onto complex 4. For two electrons moved to cytochrome C, we will pump 4 protons. So complex 3 has this remarkable ability to pump protons through mechanisms that have yet to be discovered. Okay, so let me go back. So that's what this slide represents. It represents 2 instances looking at complex 3. And we're going to get 1 copy of QH2 coming in, as indicated here, and then a second copy of QH2 coming in. And in the end, we will reduce Q to QH2, and we'll produce copy 1 and copy 2 of cytochrome C, which will move on to complex 4. Yes?
So if it's energy releasing to oxidize ubiquinol, what keeps it from just giving its electrons to anything in the matrix before it gets to this complex?
So that's a reasonable question. So it never actually contacts the matrix or the liquid in the intramembrane space. It's fully embedded in this lipid bilayer. And lipids are generally sort of an insulator. They are not particularly good at transferring electrons. You think of membrane potentials, they can be maintained for prolonged periods of time simply because they don't leak and they don't give up they don't allow for electrons to move through them very easily, or protons for that matter either. But are there other elements in that it can contact? The answer is yes, and yet the binding site is pretty specific for QH2, and in the absence of that, those electrons don't seem to come off, get donated very easily. Okay, let's consider complex 4. It has now, it has a interesting, so it's going to bind cytochrome C, not oxidized cytochrome C, but only reduced cytochrome C. It's going to come in and bind, and it's going to deliver electrons one at a time, stepwise. We're talking about maybe milliseconds between copies of cytochrome C coming in, but compared to how fast electrons can move from one site to another, let's say in complex 1, it's very slow. As the electrons come in, they go to this bi-nuclear copper complex. 2 copper metal ions present together. So they are going to be bound by histidines or sulfhydryl groups. That's what copper likes. Those electrons will move from there to a heme group, a heme A, onto A3, and then finally to a second copper center here, this copper B. So this is copper A, it's binuclear. Copper B is a single copper that is complex, coordinated, and held in place by elements of the proteins that surround us. Oxygen is also bound. And as oxygen is reduced, protons are pumped. For 4 electrons coming in, 4 protons will be pumped. For 2 electrons, 2 protons, or the equivalent 2 protons will be pumped. So let me talk just a minute about how this process may take place. So let me go back to the board again for a minute. So the cytochrome C is going to bring in just 1 electron. And we need 4 electrons to reduce molecular oxygen, which is O2, to 2 copies of water. So it's 4 electrons, 4 protons involved, but cytochrome C is only going to deliver 1 electron at a time. We have this copper A center, and we have this heme A. So these electrons move easily through these centers, and then we get to the heme A3. And so, so here's the iron that is part of this heme A3. So this is heme A3. And it is quite likely that, the oxygen is positioned and bound by both iron and the copper B. Now this is a remarkable stroke of genius because it allows the electrons to come in and to distribute themselves over all 4 of these nuclei. If I add 1 electron to oxygen, it produces superoxide, a highly reactive, almost dangerous kind of molecule, and it would be very hard to stabilize that and control that so that it didn't, this free radical species didn't interact with all kinds of neighboring species. But by keeping it, by having this sort of, this delocalization over these 4 nuclei, it allows for that to happen. So a second then electron comes in and it can move into this system. We now have the equivalent of hydrogen peroxide. A third electron comes in, we have hydroxy radicals, or the equivalent. But after 4 electrons come in, we will have 2 copies of water. We are safe, those can be released, and another copy of oxygen bound to this iron copper location within the complex that allows for the stable acquisition of electrons by molecular oxygen, preventing those intermediate species, which are highly reactive, from damaging local tissues. Now, sometimes these do get away. The mitochondria does have the ability to produce reactive oxygen species that can damage other tissues under certain conditions. Okay, so this takes us now through the electron transport chain. Again, complex 4 can pump protons. The mechanism by which that is accomplished is unknown, but it is this loss of energy as we move through each of these redox steps that is harnessed, so the energy being released as we pass the electron through, that is able to move protons against their gradient, move these positively charged protons against a positive environment in the intermembrane space. Just very briefly, it's worth considering how does the cytochrome C get in the intermembrane space, okay? It's not a large protein, but remember I mentioned that the outer membrane has chlorine channels, which will accommodate molecules up to molecular weight of about 5,000. Cytochrome C, I think, is in the range of 12,000, far too large to fit through these chlorine channels. Well, it turns out that this is just an outline of how proteins from the outside can be introduced into the mitochondrial matrix, or how they can be then moved further into the intermembrane space. And there are specialized, very highly sophisticated mechanisms, protein complexes in these membranes that can allow for these proteins to be moved. So not all of the proteins needed in the mitochondrial matrix are made by mitochondrial DNA. In fact, the majority of them are produced by the nucleus and then moved into the mitochondrial matrix through processes as described in this slide. The cytochrome C then would need to be moved into the matrix and then onto the intermembrane space, allowing it to participate in the electron transport chain. It turns out that there is evidence that complex 3 and 4, and maybe even complex 1, 3, and 4, when there is active electron transport, form what's called a respirosome, where these complexes actually they actually associate one with another. They form this even larger super complex of complexes, which may allow for more efficient transfer of electrons. Maybe we reduce side reactions of ubiquinol with other things, because now we are positioning things right next to one another and allowing for very efficient transfer. Okay, I think I will end here at this point, and we'll talk about ATP synthesis next class, which will be Monday of next week. Let me share an experience that I had. This happened many years ago when my family and myself lived in Massachusetts. We had in our ward a woman who was very active in civic affairs. She was, I don't know, pretty much every committee that they had in her town, she was a member of and was very active. She interacted with local newspapers. She would, every young man who earned his Eagle Award would have his name in the papers because she made sure that it happened. And any service project that our ward did, that was also in the news. Well, as a part of her association, she became friends with a woman by the name of Pat Purcell. Pat was, I think, somebody maybe of similar age as Polly. Polly was the member of our ward. Pat was probably a woman in her 60s. I don't think she had ever married. She lived alone and was independent, and she was blind. And one day we got a call from Polly Doggett asking if we would help. And it turns out that Pat had taken a fall. She had fallen down a set of stairs in her house and had broken her neck. And Pat now was in a rehab hospital. And Polly was asking if we would go and sing Christmas carols to her. It was that time, sort of this time of the year, maybe a little closer to Christmas. But Pat was all alone. Whatever family she had lived far away. She didn't have children. She was really all alone, and she was blind, as I mentioned. So we said, okay. My wife and I were not great singers, but we were happy to do our best. We went to see Pat in this rehab center. And it was a very hard thing to see her there. They had actually drilled holes in her skull and put bolts in, and they had these bolts secured at the top of a bed, and then they had weights hanging from her feet. So she was being stretched so that her neck was prevented from severing the spinal cord. But here was Pat. She was completely immobilized on this platform. It was miserable. She was in a lot of discomfort, as you can well imagine. And my wife and I, we spent some time there. We didn't know Pat prior to this. We felt a little awkward just dropping in and asking to sing some Christmas songs. We told her that we knew Polly Doggett, and this is how we came to be there. And so we sang. We sang our Christmas carols for Pat Purcell. And as we sang, Pat wept. And she wept because she was grateful. She was thankful. She was in a very, very dark place. Blind, completely immobilized, incredible discomfort, but was so grateful that someone would come and stand at her side and sing Christmas carols for a half hour in her situation. I mention this because I have so much more, I guess you could say, to be grateful for. I'm not in the hospital. I'm not immobilized. I can move about freely, and I have vision, and I can do many things that other people cannot. I hope that I will be even more thankful because of the blessings that I am privileged to have at this time, and that I will use my opportunities to maybe help those who are not doing well at this season. Anyway, something to think about as we move into Thanksgiving. I pray that we can be thankful and help those who may stand in need. Thank you very much. We'll pick this up again on Monday. We'll be, I guess, mostly remote, except for a few stragglers who may come to lecture thereafter. So have a great Thanksgiving holiday.